Hello, my name is Chikoti Mukuma here at UNL. I'm a PhD student and at Tamra Jackson Sims Lab. Um, I work on crown rot with Chris, who's going to introduce himself in a bit.
How's everybody doing? My name's Chris Tremundi. I'm a master's student working on Crown Rot in Tamara Jackson Zim's lab. I am also going to be the new extension instructor in Phelps County with my accountability area being Phelps, Gosper, Furnace, and Harlan counties. I begin in January.
Hey, thank you very much. We'll be talking about the research that we have been doing in crown rot of corn.
All right, so I'm going to take this first little bit, but what is crown rot of corn? Well, it is a disease that affects the crown of the corn plant, but mostly a lot of people will call it ghosting in the field. So what you have is you'll scout a field, you might notice that the plants are healthy, and then 2 weeks later you'll come out and you'll notice maybe a plant here and there within the rows is starting to turn gray green or a scalded or ghosted appearance. And then maybe a week later, you're going to come out and you're going to find that the plant has completely senesced out. And this generally happens mid to late season between R2 and R5, especially as we get hotter and drier.
Okay, thank you. So now, based on the symptomology that we have had, we've been working on crown rot for quite some time, and we have been receiving a lot of samples with the help of Bayer Crop Science. The shout out to them. So these samples have been sent to us here at UNL, and we have been trying to figure out what pathogens are associated with the samples that have been sent in. So far across the corn belt, the pathogens that we have been able to get out, most of them are from the Fusarium species. Others include those from Phytocytostroma ambigum, bipolaris, and a bit of other pathogens. Now, this is a snapshot of what we have been collecting throughout the Corn Belt. But coming to the interest that we have here in Nebraska, we find that the disease itself, crown rot, is distributed almost everywhere. We've received samples from quite a number of counties, but most of the work that we have been doing has been centered in Keith, Lincoln, Perkins, and Durham County. And from these samples, we have collected or isolated a lot of fungi, but our lab is mainly interested in the Fusarium species. So within the fungi, we have been interested in Fusarium. And within the Fusarium, most of what we are getting, which accounts for like 50% of all the pathogens or the Fusarium pathogens that we have been getting, is Fusarium graminearum, which we believe is important in this crown rot disease.
And some of you may have heard of Fusarium graminearum. Graminearum is associated with FHB, Fusarium head blight and wheat, and it is the same fungal organism that would be affecting both.
Okay.
Um, Now, when we first started this project, we were not certain when the crown rot might be coming into the plant. We were wondering if maybe early season diseases like damping off Pythium might be causing some issues with the plants later on, and we have some data that might indicate that coming up. But we did find that as early as V3, you can find crown rot as displayed in that right-hand picture there in those seedling plants. So it, crown rot comes into the crop pretty early in the season.
Okay. Based on what Chris has said, we did some trials, or we collected some data based on the trials that John Skorsky has in Lincoln and Keith County, Perkins, as well as Stewart. So what we did was to tag those plants that were looking sickly. So like what Chris had mentioned earlier, those that were showing early season disease symptoms. So we tagged a diseased plant and a healthy plant, and we tagged 100 of them, and then we followed them at different growth stages up to R5. So what we wanted to do was to see those plants that were looking that were rather sick early on in the season, what is the incidence of crown rot on those as opposed to those that were healthy? And what we found in 2024, when we first started looking at this, is that two or one of the locations in which we were working on, like Lincoln One, we had like the graph on the left side shows, we had more plants that were sick at V3 developing crown rot earlier on in the season as opposed to the healthy one. Now this was later on seen to have much of an effect in 2025 actually, where we found that in all the locations that we had looked at, there was a significant difference in the incidence of crown rot later on in the season between those plants that were showing early season disease symptoms earlier on when we did that comparison. So we see that in all of the locations, early season disease symptoms were actually a good predictor of crown rot and later on in the season.
Now, I do want to jump in on that real quick because I just thought of something. You also have to take environmental factors into account, and West Nebraska was much cooler and wetter this year than last year. So we don't know if that had an effect on this data or not, but it's something to keep in mind.
Okay, thanks, Chris.
Um, Going to out west, we work with John Skolsky out there, like I believe Cody said, in some of his hybrid trials. And in 22, he took some chlorophyll data across one of the fields out there. And here it's a little bit difficult to see, but if you look to the left-hand side of that pivot circle, you'll see a lighter green stripe running from top to bottom. If you keep an eye on that through the next three pictures, you'll notice that every week he takes a picture, the green in that strip, that hybrid trial starts to senesce or lose its color as we go through. You can go to the next slide. And in September 26 of 22, you can see it's really most of that hybrid, what they've identified as a susceptible hybrid for that area, um, is now almost completely senesced, while the surrounding corn and hybrids around it are still green, healthy, and helping with the grain fill. And then in October 3rd, that hybrid is completely senesced at that point. It's completely red. And then we have some pictures of what that actually looked like, not with color correction, but just by the eye. And you can clearly see the differences between the hybrids in that trial, with that one strip being almost completely senesced at that point.
Okay, and what we then started noticing in the fields is when we have been working is that looking at ghosted plants alone in the pairs that we have been looking at, we started finding that some of the plants that were looking healthy, actually, when you split the crown, had necrotic tissue. So this told us to say there is actually a possibility of having crown rot without having the ghosting symptom. So in the trials or in the trials that we were looking at from John Skosky, we went deeper to try to figure out just how much of those plants that were looking healthy had a crown rot. And actually, you can see what is given in the blue bars to say that there was actually a very high percentage of plants that had rotted crowns that did not have any ghosting. And when you typically just look at ghosting and you want to relate it to crown rot, it's very easy to confuse it with other diseases such as a stalk rot. That's why we believe it's more it's important to split the crown and you can verify then that there is crown rot in the plants. None of the plants that we saw had only stalk rot.
And this year was a year where there was a lot of disease pressure on plants across the state. So it's not surprising that there wasn't just stock rot in those plants this year. But what do we mean when we say split the plant to check for stock rot? Well, you dig up the plant, make sure you get an intact, at least a little bit of the root ball, knock the dirt off of it, and then you split it bilaterally down between the leaves, and you look down at the first six nodes of the plant. We've noticed when it's just crown rot, you will not see necrosis most of the time going above that sixth node, especially in those green non-ghosted plants. Um, and here is how we rate it. We take limited at that sixth node, and then we estimate the area that necrosis has taken. And in this picture, you can see we've it's an approximately about 25 to 30% of that crown has necrosis in it or that dead tissue. And then if you have a lot of plants you're rating, you could use a more simplified rating of 0 to 5, depending on how much of that percentage is there.
Okay. Yeah. Now, going back to the other thing that we have been trying to do on this project is since we saw that actually Fusarium species were very much associated with crown rot, we wanted to see how early, especially the Fusarium graminearum species complex, how early it gets to infect the plants. And so what we are seeing on the graph is that what you're seeing in red the symptomatic plants that we had tagged, and what you're seeing in blue the asymptomatic plants, so the healthy and the sick plants. So when we sampled these plants at V3, V6, VTR2, and R5, and took them back to the lab and isolated fungi from there, we found that within the Fusarium graminearum species complex, we found that inside of the plants, or rather, if infecting the plants as early as V3. But this infection looked like it remained latent until somewhere around R2 and R5 when we had higher isolation frequencies of that pathogen.
And this takes me to what I've been working on in this project is I've been trying to find a pathogenicity test or a way to test with pathogens what pathogens we're isolating would cause disease within the plant. So I use toothpicks that are colonized with these different fungal organisms. And you see with across different species of Fusarium that I have gotten varying levels of response to that toothpick into the crown of the plant. Causing necrosis in the tissue. Um, so far, mechanical wounding is the only thing I've been able to get to work, but it's possible that the plant is weakened from those early season diseases. Again, I don't have data on that, but that could be one explanation. We're still looking. And here is just some data on that. I didn't, in this experiment, I didn't get any good separation or differences between the isolates and the hybrids I used. So here I had a resistant dent, which is the orange, or what was identified as a resistant dent to me. The gray is going to be a susceptible dent, and the purple is a sweet corn, a Golden Cross Bantam, I believe, that I used as sort of a check. Again, in this experiment, I didn't have the separations, but I did get disease within the plant. I think a lot of that came down to just tweaking my experiments and refining them so that they work better.
Okay. And so in short, this is what we have been doing as a lab on crown rot. So if you didn't get anything from the conversation Chris and I have had on this, what we would want you to just understand is that what from the research that we have had, we have Fusarium graminearum species complex to be highly associated with crown rot of corn across the corn belt. And this is also seen in Nebraska, actually, because that's the main organism that we have recovered from diseased plants. Then we have also seen that early season disease symptoms in corn may predict late season crown rot. We have also seen that when evaluating corn, we have seen that not all the plants that are not all the plants that look healthy do not have crown rot. So we've seen that some plants that look healthy or have not ghosted, when you split them, you can actually see crown rot in there. So above ground symptoms alone can miss hidden crown rot infection that could be silently, of course, contributing to yield loss. And then we have also seen that field infections of Fusarium species in crown rot starts as early as V3 and tends to increase with growing stage, peaking really at R2 and R5.
Um, and that brings me to something I forgot to mention when you were talking about earlier with your data on ghosted versus non-ghosted plants and how much crown rot infection we were seeing. So the two methods can be used. It just depends on what you're doing. So if you're going to do an on-farm research and you want to research, uh, let's say, an in furrow for crown rot, it depends on how granular you want your data, how perfect you want it, I guess would be a way to
Mhm.
say it.
Mhm.
Um, to which rating you use. You can just do the ghosting, but you're going to miss a lot of plants that have the disease that look perfectly healthy. They're asymptomatic for the disease at the time, whether it be because you've had enough rain and the plant can just simply overcome the infection or whatever it is. So, I mean, both methods of evaluating crown rot can be valid, especially if you have a lot of data and your power goes up. But if you really want to get granular and specific with your data, you're going to need to come up with the amount of plants you're going to dig up, whether they be ghosted or not, and split the crown and evaluate.
Okay, thank you very much. So with that, we'd just like to acknowledge a lot of people who have contributed to this research that we have been doing. So Tamara Jackson Zim's lab, all the technicians and students, and also folks from Bayer who've been instrumental in sending in samples. And of course, the Nebraska Corn Board, as well as Bayer Crop Science for sponsoring the project, and not forgetting the Department of Plant Pathology here at the University of Nebraska. That's all that we had for you folks. Thank you very much for listening.
Thank you.
Bye.